our next speaker, we actually have two speakers. My student and my collaborator, one is Kyle Swanson and another one is Jonathan Stokes. I'm not sure which one of them is speaking first. Okay, guys, are you ready?
Yep. Okay. So next slide, please. So this is a brief history of antibiotics. I like to show this slide at the start of most talks I give because it highlights that although we've had antibiotics for the better part of 100 years now, the vast majority of our structurally and functionally unique antibiotics were discovered in a relatively narrow 2-decade period between the 1940s and the 1960s. This is called the golden era of antibiotic discovery and was characterized by high productivity in discovering new antibiotics through screening secondary metabolites produced by soil-dwelling microbes, so-called natural product mining. However, you'll see that by the middle of the 1960s, our ability to discover novel antibiotics through this approach decreased. And therefore, in more recent decades, we've turned to high throughput screening of synthetic chemical libraries in an attempt to discover new antibiotics. Unfortunately, for many reasons, the application of high throughput screening approaches for antibiotic discovery has not yielded any new clinically used antibiotics. Now, in and of itself, this would not necessarily be a problem. However, what coincides with our alarmingly lean antibiotic pipeline is the global dissemination of antibiotic resistance. Next slide. Indeed, currently about 700,000 people a year die due to drug-resistant infections. And without immediate action to discover new antibiotics, this number is expected to reach 10 million deaths per year by 2050. So in essence, we're in a foot race. Right? How quickly can we discover novel structural and functional classes of antibiotics in an attempt to outpace the evolution and global dissemination of resistance? Next slide. So if we look at our preclinical antibiotic discovery pipeline, you'll see that it progresses through a relatively straightforward path. We do a screen. In our case, we do a lot of cell-based phenotypic screens. Right? So we do a screen, we get our hits, then we elucidate the function of those hits, right? And then we chemically optimize those molecules for whatever biological or chemical property that is necessary in order to advance to more in vivo studies, right? Now, each step of this preclinical pipeline takes time, right? Luckily, earlier this year, we wrote a paper describing the use of MPNN approaches to discover novel chemicals that have antibacterial activity from really large chemical spaces, right? So using these neural approaches, we can more rapidly discover novel structures that could be antibacterial, right? That can be validated in the lab. But the downstream problem remains a significant bottleneck, right? If we have a structurally novel molecule, how do we go about elucidating its mechanism of action in a timely manner? Next slide. So even though we have a wide array of chemical and biochemical and genetic tools that we can leverage to attempt to understand the mechanism of a structurally novel molecule, this still takes many months if we're lucky and can take years or longer, right? So the logic is, next slide, can we leverage, machine learning approaches to alleviate the bottleneck that is elucidating the mechanism of action of structurally novel antibacterial molecules. So with that, I'm going to hand it over to Kyle to describe the progress that we've made so far.
Yeah, so as John said, the goal of our work is basically to build a machine learning model that is capable of predicting potential mechanisms of action of compounds against bacteria. But of course, the first challenge in designing one of those machine learning models is to actually gather the data that the machine learning model is going to be trained on. And so our first goal in the project was to obtain a data set that has diverse bacterial targets to cover as many mechanisms of action as possible so that we can predict almost any possible mechanism of action that a compound might have. And so ideally, we would try and create this data set in the lab to ensure that it's high quality. But since there are so many possible targets, the much easier thing to do is to instead mine existing databases, such as PubChem and Kimball, which already have large quantities of target data. And so as you can see in some of the statistics on the slide, PubChem and Kimball in particular are extremely large databases. They have millions or even hundreds of millions of compounds, tens of thousands of targets, mostly protein targets, but some other targets as well. And then again, millions of activity values where compounds have been tested against various targets. And so at least at first glance, it seems like these two are great resources where we could create such a mechanism of action data set. But one of the first problems that we ran into is that many of the assays that are contained in these databases actually have very few compounds. Typically, when these assays are taken from publications and put into the databases, they're not always put in with complete information. So the assays that are recorded might only have 1 or 2 molecules or maybe up to 10 molecules. And that poses a problem particularly for machine learning, because machine learning is so data hungry and requires large numbers of compounds in order to actually learn how to predict for any particular assay. And so what we've had to do instead is add certain criteria in order to filter out the really small assays and focus only on the ones that a machine learning model is most likely to be able to learn. And so in particular, we've put in the criteria that each assay that is included should have at least 100 compounds. And among those 100 or more compounds, there should be at least 20 active molecules and at least 20 inactive molecules to ensure that there's enough data for the machine learning model to learn. And unfortunately, once you've instituted these not even that strict criteria, you do lose a lot of the initial data from these databases. So instead of having millions of compounds, our data set only has about 600,000 compounds. And even more frustrating is that there are only about 46 bacterial targets across 106 different assays. And so this does cover some of the possible mechanism of action space, but it is relatively limited. And so what we've tried to do to expand our space of mechanisms of action that we can predict is actually to allow ourselves to not just focus on bacterial targets, but also to include some non-bacterial targets as well. And so when we do that, we gain another 600,000 compounds, and more importantly, a couple more hundred targets and about 1,000 more assays. And the benefit of including these additional assays, even though they're not for bacterial targets, is that very often some of these non-bacterial targets, for instance, some of these proteins might be similar to bacterial proteins, and so you can still learn something related to a bacterial mechanism of action. And additionally, there's also this idea of a multitasking benefit, whereby training a model not just on bacterial targets, but also on many non-bacterial targets, we're exposing the model to much more chemistry and much more biology. And so even if downstream we don't really care about the non-bacterial targets, the model should still learn enough so that it can perform even better on the bacterial assays that we are interested in. And so this is the data set that we've been using so far, which includes about 1.3 million compounds, 500 targets, and 1,200 assays. And the model that we've been using is called ChemProp. So it's a message passing neural network that we've developed over the past couple of years for molecular property prediction. And I won't go into it in too much detail, but at a very high level, the input is the molecular graph, along with individual features for the atoms and the bonds of the molecule. And then in the middle, the model uses a message passing algorithm along with neural network layers to take those individual atom and bond features, combine them, and then create a single molecular vector that represents the entire molecule. And then at the very end, the model uses another set of neural network layers to take that molecule vector and then make predictions for each of the 1,200 assays in our data set. And so the output is going to be, for each assay, the probability that that molecule is active against that particular assay. And so that covers our mechanism of action space. And so we've started to test our model already on this data set. And overall, the performance is pretty reasonable. We've seen about a 0.79 AUC averaged across the 1,200 assays. And so this tells us that we have reasonable performance across a wide variety of potential mechanisms of action. But there are a couple of concerning points. The first is that there's very large variability in the performance. So even though the average is about 0.79, on some assays, the model performs as poorly as 0.43 AUC, which in fact is worse than random, because if you randomly guessed, you would still get an AUC of 0.5. But then it ranges all the way up to 0.99 AUC, so it's nearly perfect. And you can see the full distribution of assay values in this plot on the right. And so one problem that we're working on is trying to improve performance on those assays where the model is currently struggling. And then another issue that we've also run into is that multitasking hasn't always worked quite as well as we had hoped. In some cases, multitasking and basically training a model across all of these assays all at once does improve performance on individual assays, as opposed to training a model just on that one assay. But in some other cases, doing this multitasking perhaps confuses the model, and it tends to perform worse on certain individual assays. And so work that we're continuing to do is to better understand how multitasking works and how we can ideally train across all of these assays without sacrificing performance on certain individual assays. But even if we build a really good mechanism of action prediction model, there's still one more problem that needs to be addressed. And the problem is essentially that when you have a model that can predict so many possible mechanisms of action, in our case, predicting 1,200 assay values, it's really hard for the experimentalist to look at those individual predictions and understand really what the mechanism of action is and what experiments they should do to validate that mechanism. And so what we've decided to do is to try to better structure our predictions using something called the gene ontology, or the Go hierarchy. And this is illustrated in the figure on the screen. And in this case, essentially what the Go hierarchy does is it's a structuring of biological space. And so at the very top, there are very broad terms such as biological process, metabolic process, that describe very general things that go on in cells, and particularly in bacterial cells. But as you work your way down the hierarchy, you get more and more specific terms. And you essentially get to the point where these terms represent meaningful mechanisms of action. But the nice thing is that the terms are still general enough that they can cluster a bunch of these individual assays into one term. So for instance, protein splicing might be relevant to 10 or 20 different assays. And so instead of having to look at 20 different values, you can look at just one single prediction value for protein splicing. And so what we've done is we've actually built a custom web visualization tool, which is able to take the raw assay prediction values of the machine learning model and then convert them into these Go predictions. And so you get something that looks like what you see on the screen. So in this case, it's showing just a small section of the Go hierarchy and some of the predictions that the model has made for these Go terms. And you can see in particular, we're highlighting the protein splicing Go term. And on the right, you can see all of the individual assay predictions, which are then summarized into the singular protein splicing term. And the really nice thing about using a visualization tool like this is that it makes it very easy for the experimentalist to quickly look at the Go hierarchy, see clusters of biological space that seem to be most affected by the molecule, and then perform the experiments that could validate those potential mechanisms of action. And then the final step in the process is actually doing the experimental validation to see if these mechanism of action predictions do in fact work. And this essentially requires two main steps. So the first step, even before we can think about mechanism of action, is that we need to find compounds that do in fact inhibit the growth of bacteria. Because if the compound doesn't do anything to the bacteria, then clearly there's no interesting mechanism of action. And so to find those compounds that have inhibitory effect, we actually train a separate ChemProp model on whole cell inhibition assays. And we've already trained this model and made some predictions on a set of 800,000 compounds from the Broad Institute. And we're currently in the process of testing the top 300 compounds in the lab. And then once we found the handful of compounds that do have inhibitory effect, then we can turn to this mechanism of action model to make predictions for the mechanism of action of these compounds. And so the steps involved there are first training this ChemProp model on all of the target assays that we've collected. Then we can make mechanism of action predictions on the compounds, use the web visualization tool to interpret and understand those predictions, and then finally go into the lab and perform the experiments to validate those predicted mechanisms of action. And so to quickly summarize what we've talked about, we've said that determining mechanism of action, at least using current methodology, can take months to years. And we're hoping that machine learning can greatly accelerate this process. We've also seen that databases like PubChem and Chemble contain large amounts of target data, even after filtering out some of the smaller assays. And so they're great sources of training data for these machine learning models. And then once you have all of that data, we can now build machine learning models that can predict thousands of possible mechanisms of action simultaneously. And then finally, we've talked about how we can use visualization tools to aid in the interpretation of these machine learning predictions, which can then guide experimentation to more quickly identify the most likely mechanisms of action. So thank you very much, and we'd be happy to answer any questions.
So thank you very much, Kyle and John. And I just want to answer a question that some have in mind, why we're talking about antibacterials during COVID conference. There were multiple studies, and maybe John can comment on it briefly, that demonstrate that many of the COVID patients that are hospitalized actually suffering from the bacterial infection. John, do you want to say one sentence on that?
Yeah, I think, I forget the precise statistic. I think it was something like 50% of COVID-19 associated mortality was due to a secondary bacterial infection, right? So this COVID-19 pandemic, particularly given the increased antibiotic use in severe COVID patients, is going to have a knock-on effect on the evolution and dissemination of resistance, right? So that's kind of the link between the COVID-19 pandemic and the downstream wave of antibiotic resistant infections that we may see.
So John, and for you, there is a question from Pat Walters. I think it's for you. In many cases, teams have been able to design antibacterials that can inhibit a bacterial target. Unfortunately, these compounds are ineffective because they are influenced by wild type bacteria. Will your model capture influx?
So the way that we're setting up the antibiotic selection process prior to mechanism of action determination, we'll select for compounds that can both get into cells, particularly gram-negatives that are difficult, that have a high permeability barrier and are not effluxed. Importantly, though, that's a good question. We've talked about this a lot before. How can we develop models to predict compounds that either have excellent gram-negative permeability barriers and or very low efflux rates? And it's feasible, you know, we have genetic tools such that we can engineer bacteria to gather training data to be able to do that, yeah.
Thank you. And a question for Kyle, this will be the last one, and then you guys go and answer the questions in the chat. Okay, do you treat each assay as an independent target? It's for Kyle, or can you use distance in a Go space as a way to structure the loss function or representation space?
Yeah, that's a good question. So in the current model, we are actually treating each assay as a separate independent prediction. There is still some sharing of information because it is one message passing neural network that is making all of the predictions for each of the different assays. So there is a lot of information sharing within the model, but at the very end of the model, it is still a totally independent prediction. Actually, in the past, we had experimented with trying to make predictions directly on these Go terms and actually leveraging the interactions between the Go terms and the structure of the Go hierarchy. But we weren't able to basically improve performance at all by including that additional structured information. So for now, we're just sticking with the simpler model, which is independent predictions for each individual assay.
Thank you. Thank you very much, Kyle and John. And